All right, so we can start. My name is Jerry Rosario. I am one of 3 ambassadors for India of the Usher Syndrome Coalition. So Usher Syndrome Coalition is an international organization, and there are more than 2,000 members worldwide, families with Usher, individuals with Usher, children, adults who are members of the Usher Syndrome Coalition. And the goal of the coalition is to connect those families with Usher syndrome with information, with researchers such as Dr. Kumar, and with each other, and with the rest of the community. So we are very lucky to have Dr. Kumar here. He is from the Narayan. I'm sorry, I am not. I cannot pronounce things properly, so I will let Dr. Kumar introduce the names himself. But he is one of the top 3 hospitals here in India, top 3 eye hospitals here in India. So without further ado, I'm going to give it over to Dr. Kumar. Oh, by the way, I'm sorry. Can I request everybody to mute yourself? Okay. If you have any questions, okay, maybe we can have a break for questions. It's up to Dr. Kumar when he wants to give a break for questions. Okay, clarifying questions, and then the rest of the questions towards the end. We have about almost a little over an hour for this talk, but we'll definitely have question and answer portion. And when you do have a question, first please identify yourself, say your name, so that the interpreters can also identify you to the rest of the attendees. All right, thank you so much. Dr. Kumar.
Okay. So thank you, Jerry, for asking me to start with the talk, and Nancy from the Usher Coalition Group for giving me this opportunity on this evening, Indian time. I will start with my talk, and I will introduce myself once I start. Let me share my screen. So I share my screen. I also make it a point to check if everything is okay. I hope my screen is visible. Everything okay, Jerry?
It seems to be okay. All right.
Okay. So the talk that I'm going to give is on Usher syndrome. It will be an update on the genetics, genetic counseling and therapies, and also very briefly touch upon on the clinical aspect so that everybody is on the same page. To introduce myself, I'm Kumaramanickavel. That's a long name, so the easy way to address me is by calling me Kumar. And I worked and studied in India, did my MBBS, and after which I did my MD. Having finished my MD, I went to New Zealand and worked with Dr. Michael John Denton, who was the leader of the ocular genome group in the University of Otago, New Zealand.
Excuse me?
Yeah.
I'm sorry to interrupt. Somebody said that the audio is low. If you can please speak a little louder.
Okay.
Thank you.
So I went to New Zealand and I worked with Michael John Denton and his team. And over a period of 5 years, we mapped almost about more than half a dozen genes that cause various types of vision-related diseases, particularly affecting the retina. And one of them is RPE65 gene that has come into therapy today. So that we identified in 1996 and published in 1997 in Nature Genetics. And then I was at the National Eye Institute, National Institute of Health, Institutes of Health in Bethesda in the US. And I worked with Fielding H. there, the ocular genome group. And I came back to India, worked in Shankar Nethralaya, a premier eye hospital in Chennai, southern India, which was headed by Dr. S.S. Badrinath. And I set up a genome department there and also a genetic clinic in the same hospital. I left that hospital in 2009 to start more such genetic clinics and genetics departments and ocular research in India. And I moved to Narayan Nethralaya in Bangalore, where I still work. And I also help institutions in Asia and collaborate with groups in Hong Kong, China, and a few other places. So with that introduction, let me get back to my talk. And today I'll talk about Usher syndrome. And we do see many of our patients, and I will use only the term subject and not patients hereafter. And these subjects come to us with various ocular related diseases and have their own issues for which there isn't any kind of medical help. So when there isn't a medical help, that doesn't mean that they don't have any solution. And I'll come to these problems that they face in life where we try to give solutions in genetic counseling. So before I get to that, let me move on with my slide. And I sort of divided my talk and structured it in such a way that I talk about Usher syndrome first, then genetic counseling and how we help people, not only in Usher syndrome, in other conditions too. And then finally, I'll come to therapies. And therapies is of a very high importance, and therefore I have made it as a very brief talk because it's very technical to be given to a very lay audience, and therefore we'll discuss a lot about therapies in question session. We have many sense organs, which is a very, lay term, right from our eyes, skin, tongue, nose, ears, and so on. And each of these areas of sensation, we have nerve endings, which we call as receptors. And these receptors help us to, have the taste or hearing or vision, touch, and so on. Now, these receptors are very important, and the receptors that we have for the eye, particularly in the retina, is the rods and cones. We'll come to that subsequently. Now, eye is a very dominant source of sensory perception of the external environment. And we can appreciate various kinds of colors as listed in the slide over here. And we have a very high resolution using our eyes, about 576 megapixels. And our eye muscles are very active muscles, very, dynamic in nature of all the muscles that we have in our body, and we use this the most. Okay. Now, if you look at the cross section of the eye, you take an eye as a ball, and then you try to cut it across. This is how your eye will look like. Starts with the cornea, and then you have the pupillary opening, and then you have the lens. Lens and cornea are transparent, so the light moves from the cornea to the lens and through the eye and hits the most posterior or back end of the eye ball and reaches the retina. And then the retina is stimulated, and that information goes to the brain. I'll come to that in detail in the subsequent slides. So when an ophthalmologist looks into your eye, I'm not an ophthalmologist, I'm a geneticist. And therefore, when an ophthalmologist looks into your eye, this is how your retina will look like. So when you look at the retina head on, then this is how it looks like, and you can see this structure, which is a yolk-like in appearance, and that's where the arteries and the veins get in and get out of the eye and supply the nutrition and remove the toxic products and so on, depending on whether it's the artery or the vein. And to the left or to your right, you can see that you have the a slightly dark red portion, which is called as the macula. And this macula contains most of the cones. And these cones help us to read and look at in bright light, say, for example, sunlight, daylight, and also appreciate colors. And these cones are present in the center part of the retina, and the remaining part is fully filled up with rods. Okay, rods help us to see when the light is very less, so it helps us to see the contours and the shades that are there, but they can't appreciate color. Now, if you look at cut that retina, okay, and then look at the rods and cones, right? They will appear like this if you magnify them, okay? Now, if you look at the retina per se, it has about 10 layers, and each of them are very critical in the visual function. And the most important part of the retinal layers is the sensory perception layer of rods and cones. So a rod looks like the way, as you can see here, and the cones look like this. So because of the shape, they have got the names. And they have two distinctive functions. As I said, dark vision or less light perception is by the rods, and daylight or color vision is by the cones, and they are in the center part of the eye. So therefore, when you have conditions like retinitis pigmentosa, the rods are the first affected, and therefore, since they are present in the periphery or in the sides of the retina, you start to lose the peripheral vision. Whereas when the cones are affected, for example, in Stargardt's disease, when you have reading disabilities and you can't see images very clearly, and the central vision is lost, and that's the distinction between rod degeneration and cone degeneration. So, as I mentioned, rods help you to have this black and white vision in dim light, and cones help you in color vision or bright light. And we have about 120 million rods and 6 to 7 million cones in each eye. So that's the accountability of these rods and cones. Somebody has to go on mute, please. Okay. Now, when the light strikes the retina, particularly the rods and the cones, then there is a chemical reaction that is immediately triggered, and it's called as the phototransduction pathway, and that happens in the neuroretina. And there are a lot of end products out of this reaction, and that is completely cleared by the retinal pigment epithelium, where there's another reaction that takes place, which is called as the visual cycle. Okay, phototransduction and visual cycle. So therefore, when phototransduction happens, we lose a lot of these chemicals, and these chemicals are recycled in the visual cycle by the retinal pigment epithelium, and they are again thrown back into the retina for recycling and reuse. Okay. Um, why am I bringing this point is that there are more than about 300 to 350 proteins and structural and biochemical proteins, and each of them is represented by a gene. Okay, so rhodopsin is one gene that is present in the rods, which is triggered by the photon, and similarly there are another, 349 genes and proteins, responsible proteins, which are involved in the phototransduction and visual cycle pathway. So therefore, the conditions that affect the retina or the retinal pigment epithelium in either the phototransduction pathway or the visual cycle are not easy to be solved. And therefore, the therapies have become a very big challenge in the field of medicine, and particularly in ophthalmology. Now, the second component of Usher syndrome, the first component is the visual component of the condition, and the second component is the hearing. So, in the hearing, in the ear, we have three parts. One is the external ear, which you can see here as the ear pinna and the ear canal, and then you have the eardrum or the tympanic membrane. And the tympanic membrane vibrates as I'm talking to you, and you're hearing my voice as sound. And when the tympanic membrane is shaking, there are three different very small bones attached to it, and that is present in the middle ear. And these bone, the last bone, the third bone of that system is again connected to the inner ear, and in the inner ear, it is connected to a part called as the cochlea. So it starts, one bone starts at the, as I can show you in the cursor here, starts at the tympanic membrane or the eardrum, and then it goes and connects to the cochlea. And this is called as the staples, which connects to the cochlea. So cochlea is something, the shape is almost like a snail, and it has labyrinths, and it has a fluid, and that fluid is the cochlear fluid. And when this stapes is moving, it starts to create a vibration, and you have sensory nerves in the cochlea, and these nerves start to move. The receptors start to move when the staple starts to shake. Okay. Now, as far as the retina photoreceptor and the hearing receptor, they have certain structures which are hair-like structures or ciliary structures, which they are called. And the proteins that are there in the eye ciliary structure and the hearing ciliary structure are similar in nature. And that is why when you have Usher syndrome, it has about nearly 14 to 16 genes that are causative of Usher syndrome. Then if one mutation happens, then it affects the ciliary part of the photoreceptor as well as the cochlear receptor. And therefore, when you have one gene affected, it affects two parts of the body, and therefore people with Usher syndrome have both visual as well as hearing conditions. Okay, right. So if you ask a common man, how do we see and how do we hear in terms of a sense of perception? Then always everybody says that I see with my eye and hear with my ear. Okay. In medical terms, that is not right. Okay, in physiological terms, the eye and the ear are tools, and so is the tongue. Okay, and every other sensory organ. They're all tools that we use, and that tool helps us to perceive the information. And once the information is perceived, it's sent to our brain, and the brain tries to get that information and then analyzes it and then gives it as vision, hearing, taste, smell, and so on. Okay. So the region that helps us to see is actually the back part of the brain, which is called as the occipital lobe. So that's the region which helps us to process the information that is provided by the eye. And the temporal lobe, which is over here, is a region that perceives information or receives information from the ear cochlea and then interprets that as sound. Okay. And the whole brain, there is a region called as general interpretation area that connects both the vision and the hearing and makes us to understand things. Okay. For example, we see the train for the first time in life, and our mom or dad tells us that that's the train. And when the train makes a sound, then we connect that sound with the train. Okay, the sound with the visual perception of train. And anytime we hear the sound alone, we know that it's a train. Okay, so that kind of interpretation is done by the general interpretation area. And so it goes with taste, any food, a smell of the food. You don't need to see it to tell what food it is. So therefore, the brain is the chief organ of our all perceptions. Okay, sometimes when people get injured in the occipital lobe, they can become blind even if the eye is perfectly normal. Okay, so we now know that why people with Usher syndrome have both hearing as well as vision related problems. Okay. Now, since I told you about 350 different genes are involved in phototransduction as well as visual pathway, you can compare it with a car. Okay. So when you put your key into the car and then you try to start the car, the ignition starts and then you have fire that comes out of spark plug, and then you have the gas that gets into the engine, and then you have combustion, and the combustion creates a pressure, moves the piston in the engine, and the piston moves the crankshaft, and that shaft moves the tire, and the tire starts to move as you accelerate and apply for the gear, or if it is an automatic gear, and so on. So the point at which you put your car key and start, and to the point where your car starts to run, there can be more than, a couple of hundred reactions that take place. Each and every step is very important. So therefore, putting in the key is equivalent to photon striking your photoreceptor. And then your visual perception at the end is sensed, and similar as the car's moving response. So therefore, if your carburetor doesn't work in your car and reactions, one, you put your key and everything will start to, fire and move. But then in the carburetor component, which can be the 75th step, doesn't work, then the whole process stops at 74. Okay, it doesn't cross that and go to 350 in the eye. So what do you do then? Then what you have to do is that you have to replace that 75th gene, okay, by a gene therapy, or there are other therapies. We'll come to this at the end of our talk. So that is how the scientists and doctors are trying to solve the genetic diseases problem. Okay, they're trying to fix so that these pathways are completed, and therefore at the end of it, the subject is able to have or recover or rescue with their vision as well as hearing. Okay, so we'll talk about that as we come to the end of our talk. Now, I have actually completed talking about what is Usher syndrome, and then I will move on to the next part, which is genetic counseling. I'm using some of my patients' data here. I have taken their permission, but nevertheless, I have kept it very private. Uh, I have to keep my ethics, okay, in not trying to reveal too much about what I'm talking and telling about uh, their condition over uh, in an open talk. We do use this data a little more openly without revealing people's privacy, okay, identity in our medical talks. But I can't do that when I'm talking to sort of an Usher syndrome group, okay. So some of my slides are quite uh, concealed, uh, but then I have to use them when I give a talk and somebody wants to know more details. I will definitely reveal it, but without revealing the privacy of my subjects. Okay, so what you see here is that of one of my very long time subject who's been with me for quite a long time. And he has the condition called as Usher syndrome, as we are talking. And his retina is what I'm showing here with quite a lot of pigmentation. And his daughter also picked up some of the information of her is also here in the slides. And this is a bit about their family tree, and he and his daughter are affected, and others are not affected in his family. And when we ran through some of the genes that causes Usher syndrome in his family, we found that he has USH2A gene that is having variants, and that is causing the condition. So there are two variants in his family, and one of the variant is common between him and his daughter, and the other variants are different between the father and the daughter. So I'm quite trying to help him to get into uh, the Usher syndrome natural course of the disease um, trial that is going on in the US, and he's now enrolled into it. And he's very much on therapy because of his financial condition or background, I would say. And more, he's more concerned about his daughter. Okay. So when we do this genetic counseling, it's not only that we are just vomiting out the genetics to them and we end up with that. That's not the way we do it in India. We get very close to them and we are very empathetic, and we try to explain to them each and every aspect of uh, progress of research and what is the current status. Okay, so we talk about the genetics so that they know why it just happened in them. And then we talk about a bit on the natural course of the condition and how it can lead and proceed further. And after which we come to the genetics part and so on and so forth. Next to this stage, there is a stage where people get inquisitive about getting married. Okay, that's going to be the next subject I'm going to talk about, and for one of the reasons why they come. So in India, people come for four reasons. Okay, and I'm sure that is the condition across the world. The first reason, I think I have to go to my next slide, but and therefore I'll come why they come for counseling in the subsequent slides. So Usher syndrome, as I told you, has more than a dozen genes involved, and you have you know USH gene, which are of different types. Okay, and USH2A is one of the most common condition that we see in India. Okay, and across the world too. There is a catch in this gene. It's because it's a very big gene. Okay, so therefore, when people do gene therapy, briefly about gene therapy is that they take the gene of interest and they pack it inside a virus. Okay, like the way COVID gets into us. So what COVID does is that it has its own genome, and then it sort of pushes itself into our cells, and then it gets into our genetic machinery of the cell, and then it starts to reproduce in that machinery, okay, and multiplies itself. And then, we get affected with COVID, and then we spread with coughing and, breathing along with the other people who are not affected. So the virus from us sort of gets transferred to the next individual and keeps on passing over from one to another and becoming a pandemic. Okay, so that's the beauty of virus. So it can get into our system, multiply in our system, reproduce, and then they can get out and then go ahead. So scientists captured that ability of the virus, and therefore what they did was that instead of their genome, they they inserted our genome into it, our gene of interest into it. Okay, not the entire genome, but our gene of interest, and then they made that gene in that virus to be reproduced. And now we have, say, for example, USH2A gene that is defective being produced. We can produce the right copy of the USH2A inside our own cells. It can be the photoreceptor cells in the retina, or it can be the cochlear cells in the cochlea. Okay. So, cochlear gene or the protein in the photoreceptor or in the cochlea. Okay. So therefore, that is how gene therapy works. But again, packing a gene of H2A size is not easy. Okay. Various other conditions of retinal degenerations like RPE65, which causes labor congenital amaurosis, a very severe condition, early onset in nature, which does make a young child lose vision. Whereas Usher's is a very slow one. It is late in onset in terms of visual problems, and it's not as severe. Okay, it is slow and later in onset Usher, but then labor congenital amaurosis is much earlier. So therefore, IP65 is comparatively a smaller gene. It can be packed inside one virus, and that can be made to be multiplied in a laboratory, and then you can transfer that, inject it into the retina by subretinal injection, as it is said. Very small volume is enough to have millions of viruses of our interest. And these viruses are basically adeno-associated virus, which is a common cold kind of virus, and they are made very safe, okay, before they are made to be infected in the retina or injected into the retina. So Usher syndrome still struggle in terms of gene therapy, and now they are doing a gene therapy in Europe. Okay, there were two groups. One which was in Netherlands called Procure. Nancy knows them more better than I do. And they have moved that study, which is an exon skipping study. I'll tell you what that is. And then another company called Sepal Bio has now taken it over. So what they did was that, so when a gene, if you say a gene, the exact way to explain what a gene is like having a train with many compartments. Okay, that's how a gene is, and these compartments, which we call as exons, are amino acids, and then from that exon's amino acid, we get the whole protein. So it can be a rhodopsin protein, can be insulin, or it can be estrogen, or it can be growth hormone. Okay. So that is how they are produced. So they are basically genes, DNA, which is translated into, transcribed into mRNA. Okay, a copy that comes from the nucleus into the cytoplasm of the cell. And it's a transcribed copy. It's from a DNA to RNA. And this RNA is translated into a protein as amino acids. Okay, and they get out of the cell and do the function, or they could do the function within the same tissue. Okay. So these are like compartments. And you can have a wrong DNA copy in one of the compartments. The compartments are about 100 in nature. You can have the 15th compartment a mistake. Okay. Now in exon skipping, what they do is that if you have a mistake in the 15th compartment, they look at it in such a way that from the 14th compartment, instead of going to the 15th, they jump straight to the 16th. Okay. So they make a protein without that 14th compartment, but still that protein will be fairly functional. That is exon skipping. And this is something that ProQR has been doing in Usher's syndrome, particularly in USH 2A. And I think the pandemic came and created quite a lot of hardship. And this now is taken over by a French company called Settle Bio. So therefore, gene therapy is fairly a successful way of treating people with genetic conditions. I'll come to that at the end of my talk. I'll show you a video to believe that, okay, of gene therapy. Now, this is a slide showing that in Usher syndrome, this is the eye, and then, so the photoreceptors are affected. And then it's also the cochlea where you have another type of a cilia which is also affected. Okay. Now I'll go into the genetic counseling per se of my talk. It's part of my title today. So patients come for four different reasons as shown in boxes over here. So they come for clinical diagnosis. Okay. So at times when your clinician is telling you to do a genetic test, you should ask, why should I do the test? Okay, everybody should not get a genetic test done until otherwise you fall into these four categories as shown in this slide. Now, when the clinical diagnosis is not clear, say when you say retinitis pigmentosa and when you say Usher syndrome, they don't sound the same. Okay, because retinitis pigmentosa is a condition which affects the retina alone. But Usher syndrome is a condition that affects not only the retina but also the cochlea. Okay, so when a condition affects two different parts of the body, or more than two different parts of the body, multiple different parts of the body, then we call that as syndrome. Okay, sometimes what can happen is the one condition might be diagnosed because a person is a specialist in that. Okay. So somebody might have retinitis pigmentosa with mild hearing difficulty. And they can actually fairly hear, and they start to also adapt by having the ability to lip read. So an ophthalmologist might easily miss the hearing component and diagnose it as retinitis pigmentosa. Okay, it's not uncommon to happen, and can't blame the clinician for that, because in India they see 60 patients in one day. Okay, and sometimes more than that, some of the senior physicians. So it is easy for them to miss such kind of problem when the subject is not telling you they have mild hearing issue. So therefore, when they do a genetic test, they will know that it is, USH2A or USH gene that is causing the condition. Okay, and they can correct themselves. It's not an isolated problem, but it's a syndrome. And also knowing that what type of gene is affected, whether it is autosomal dominant, autosomal recessive, or X-linked recessive, they can predict the natural course of the condition. Say in autosomal recessive or X-linked recessive, the condition can be deteriorating very quickly. So they have to be sent for appropriate rehabilitation so that they're able to cope with the situation much faster. Autosomal dominant is generally much slower. Okay. And not only that, it has a lot of implications about the marriage and next child and so on. And therefore, I move from the first box to the second box. So in the second box, people come to us to know whether they can get married because they have somebody with a genetic condition in the family, or they have immediate relative, like a brother or sister affected. Will that cause any issues with them? Or they themselves might be affected, and therefore they can ask, "Well, can I get married and have normal children?" And or they can come and say, "Look, I would rather prefer whom I should choose as my partner." Okay, and there can be people belonging to a particular group, okay, or a community where a genetic condition might be much more prevalent. Okay, for example, if you take uh, in the US, sickle cell disease is quite predominantly present in the American blacks. Okay, and therefore in New York it's mandatory that they have to undergo genetic counseling because two people might be carriers of sickle cell disease. Okay. And the the third reason why they come is that they have a previous child, okay, and or they might have a family history and they would have been already married, and therefore they come for preconception counseling or prenatal genetic counseling. Okay, so and there are many tests to be done prenatally, and you can test them and show you one of our patients in whom we did do prenatal diagnosis. And the fourth reason is the Usher syndrome 2A subject I showed earlier. People come to know of research, intervention, genetic research, gene therapy, stem cell therapy, you know, retinal implants, okay, clinical trials for a particular gene and so on. So therefore, they come and want to know what gene it is. Okay, so we clearly tell them these tests will not give you any result or benefit after knowing what gene it is. Okay, so you know you have Usher syndrome. Doing a genetic test and knowing molecular diagnosis of which is USH2A is not going to benefit. Okay, we don't do a genetic test to know molecularly what it is. Okay, you're only trying to recoin somebody's condition. So otherwise, for the first three reasons, when they come, we straight away go ahead and do the test. Okay. The fourth one, we make them very clearly understand that it's not going to be of any benefit doing this test because gene therapy and other things are not still in the pipeline of prescribing uh, practice. Okay. Right. Now let me go to, I'm not going to go all of them and go to the second one straight away in a premarital situation. I masked the slide, the pedigree. So here's a girl who's, in mid-20s, graduate, who whose parents married consanguinely, they were uncle and niece, which is not uncommon in our country, particularly in southern part of India, where such blood-related marriages are not uncommon, but that is, dwindling very fast with education and knowledge and awareness. And these parents are about, close to 50 years of age, and therefore half a century ago they got married consanguinely. And because of that, she's affected. Okay, and she has her own other sibling unaffected. Now, because she's a graduate, she read somewhere and knew that it's a genetic condition, it can recur in the family. Therefore, came for counseling. We took the pedigree, it was classical autosomal recessive inheritance. And we told her clearly, if you marry outside your family, outside your community, then your children won't be affected. But being a graduate, she put her foot down and said, yes, I know what you say, but then I want to know exactly what my gene is, and I want to know what the mutation is. Okay, so we did the test, and when we looked at a gene, we found a particular gene which is a bit more common in India to be causative of Usher syndrome. So she had a GPR98 gene exon 70. This exon 70 is the compartment I said, where she had a change from DNA letter C to T, a glycine in the 4777th region became a termination, which means it stopped at, you know, translating amino acid. And it was in two copies because the parents were blood related. They gave the identical copy to her, and she had Usher syndrome type 2C, autosomal recessive, and the classification of that variant was pathogenic. And somebody wants to know more about it, I can talk in the question session. What is pathogenic uh, or variant of unknown significance and so on. Now, so therefore, it's the same her pedigree, and we told molecularly that, see, look, you have two copies wrong, and when you get married to somebody who's not going to be blood related to you, they won't carry this copy, but we'll check it before you get married or after you get married, and then we can be sure that the other partner, is not carrying it, and you will definitely give one copy to the child, and your partner will give one right copy. You will give one wrong copy, you'll give one right copy. A child needs two wrong copy to have the condition, and therefore your child will not be affected. Okay. I don't know whether she got married or not, but she was quite satisfied with what we said. Okay, this was a couple of years ago that it happened. Now, this is something as solution we give. Okay. Even many clinicians think that the moment somebody has a genetic condition, that means that nothing can be done, and therefore, of course, from treatment point of view, what they say is right. Okay. Today, there's nothing that can be done, but tomorrow, research will definitely give you a solution in terms of treatment. But today, the genomic knowledge can give you many solutions. As I told you, in trying to know what syndrome it is, the first option. The second option as marriage options. What can be the option that you can choose? Okay, you have the power. I think, unlike the way we do medical practice, the autonomy in the medical practice is with the medical doctor or the physician, because they decide what test to be done, what surgery to be done. But in genetic counseling, the autonomy is with the subject and their family. Okay, they tell us what we should do as genetic counselors. Okay, we don't have that autonomy, and that's how the guidelines are set. The American College of Medical Genetics guidelines and most of the other countries' guidelines are set in such a way that genomic practice, the autonomy is with the subject and the family, right? Now, I'll move on to the preconceptional family that we counseled. And of course, these are not Usher syndrome. Okay. The previous condition was Usher syndrome, but not this one. So this was,
Excuse me, doctor.
yeah.
I'm sorry. Um,
Yes.
can we stop for a while somewhere and have others ask questions if that is possible?
Yeah, go ahead. I'm happy to stop, and then they can ask questions, and then I can continue this again. Not an issue.
All right. Because I'm seeing some comments in the chat. I believe some people were not able to follow properly.
Okay,
And also just okay, somebody had mentioned if you can stop sharing the screen, if you don't have if you can just talk through the topics, because some people are not able to see the screen properly, as
okay,
well as they're not able to see their interpreters properly because the interpreters are now small sized.
okay. Okay, you can switch over that my screen to be a smaller screen and then the interpreter to be larger. And I can take questions now.
Unfortunately, I cannot control the size of the screen sharing.
Okay.
Anybody has a comment? Questions? If none, we might be able to proceed. Let me ask the ISL interpreters, do you need to switch?
Yes, now it's
Okay.
about 30 minutes, so we will switch. Arva, just replace as a spotlight.
Okay, I will remove the spotlight from Arva.
And Jerry, Yash, this is Nancy. Yash is signing. It looks like he has a question.
Okay.
Saurabh, can you see Yash?
Okay, so he has signed out that you have explained. We learned everything, but like in deaf or in hearing, there are some genetical issue. Or I want to ask that if a deaf person in a deaf community,
Mhm.
okay, he want to know about deaf community and sign language, like hearing person have syndrome or syndrome. You are talking about regarding to the hearing community, right? As any deaf person have faced this issue, any deaf person,
What issue?
Usher syndrome.
Of course, actually. It depends on where, what kind of institution it is. If it's an ophthalmic institution, people come with vision problem and then the hearing condition is diagnosed. Whereas if you're running a hearing institute and the hearing issue is detected and they go to a hearing institution and then finally they know that it is Usher syndrome. Okay, both ways it can happen. Okay. Both conditions don't happen at the same time. I've seen it with my subjects. So the vision problem can, usually it is later in onset. The hearing condition is.
Okay, for example,
Hmm.
in world in laboratory,
Mhm.
there are a lot of changes. There are a lot of like
So
on
I,
visual and visual chemicals
no,
and laboratory.
your voice got lost in the middle.
I'm audible?
Yeah, you're audible, but your voice got lost in the middle. If you can repeat what you said, that would be good.
Okay. He's asking you that how we can improve visual impairment. The
Um,
basic question is how we can improve any visual impairment in Usher syndrome.
okay. See, there are visual rehabilitation programs. So what happens with visual perception loss is that, as I was telling there are 120 million rods and 6 million cones in each eye. So when you start to lose the rods, you start to lose the peripheral vision, and you start to lose the, uh, twilight vision or low light vision. Okay, you get into a movie theater, you find it difficult to adapt and get into the seat or in your the row that you're allotted. So therefore, they slowly also start to lose the reading ability because the cones also start to deteriorate. So when the cones start to deteriorate, or when the rods start to reduce and your peripheral vision is getting reduced, then when the peripheral vision is getting, tubular and tubular, they can give you certain glasses that will help to widen that view. Okay, that's the rehabilitation when your rods are getting lost. Whereas if your cones are lost and your reading ability is getting lesser and lesser, they try to give you certain lens that will magnify, or they can use certain um, say devices that helps to magnify. If they are reading a book, there can be some kind of a desktop which will magnify, or it can be your mobile phone that can magnify the letters so that you can actually read better. Okay, so that is how they try to rehabilitate people as they slowly start to lose the rod or the cone vision. Okay, that's a field by itself, and most of the eye hospitals in India do have those services of visual rehabilitation. And usually the optometrists are the people who deal with it, and there are quite good devices, wearable devices, okay, which you have very good companies across the world, which provide you with certain wearable device that you can wear in your spectacles, and they can widen your vision or they can magnify what you're seeing and so on. Okay, so I hope I have answered that question. Okay.
He's asking that when we're getting elder, so our vision become weak, right? So I want to ask that how is it possible when I am in 80s or in 90s, my vision still stays strong. I can see clearly without any issue. So how it can be done? Because now our lifespan become less and our vision is not good in 21st century. So I wanted to know that how I can improve my vision. And in some deaf community,
Mhm. See,
people don't know about us syndrome because they are just staying at home. They don't know about vision impairment and all.
okay,
There is lack of accessibility. So how we can sort it out?
okay. I'm not expert in hearing disability per se, okay? But I know that if you take 100 people with hearing disability, only a small section of people will have Usher syndrome, okay? So sensory neural loss, deafness, there are more than 100 genes responsible causing defects in the cochlea, particularly, okay, in the inner ear. And Usher syndrome is a very small section of that, okay. But Usher syndrome, if you take in terms of uh, visual conditions, Usher syndrome is quite a big group, okay. And therefore, if you take hearing per se, there are genes that cause only hearing disability. And those people will never get any visual perception loss. Okay. So we should not get confused between hearing. Everybody who has a hearing problem will develop Usher syndrome. That's not the case. Okay, so there are several genes that causes hearing problems, and they don't cause vision related issues at all. Okay, yes, there's some question from Divya, I think, right?
Hello, this is Jerry. I think we've lost the ASL interpreters. Who should I
This is the
spot?
interpreter speaking. You have not lost us. I
All
just
right.
need you to spotlight Diane, please.
All right, got it.
I think Divya started off with some question. We can probably, she had
Yes, Divya does have a question. We're just waiting for her to be able to see the interpreters in order to ask her question. Please just hold a moment.
Diane, you are spotlighted.
Thank you. My question is, what does the future generation of gene therapy look like? As what is the plan for the future generation of gene therapy?
okay. I've come to the component. I've not yet come to the component of gene therapy. I'll finish my talk and then talk about it, or do you want me to talk because it's a little premature at this point of time. I will complete hopefully another 5-7 minutes. And then shall I talk about that? Or do you want me to talk right now?
This is Divya's mom,
Okay,
Dr. Kumar.
all right.
The problem with your, in no way do I mean disrespect in any way.
Okay,
This is too scientific for everybody. Perhaps if you had done a small, like a presentation, but a less scientific presentation for everybody, and just points is what we needed at
Mhm.
this time, so that we can grasp what you're trying to tell us
Okay,
and then ask proper questions. This is much too scientific.
okay. I have not done too many, talks to people with the condition. I've done it in India. This is the first time I'm doing it online. I have kept my talk very low tech. I have not kept it very high tech. In fact, that's how I felt. All right. Otherwise, it will be, uh, I don't know if it will be, if we don't use the medical terms and give a talk, it will not, because I've been trained in US and in New Zealand. I can't give a very, very superficial talk. Okay. Certainly, we can, if you feel that my talk is very scientific, you can note down points and you can ask me those questions where I've dealt it very scientifically. I can make it quite simple. In fact, that's why I put a car engine as a simile and try to explain it.
The other issue that we have with this presentation is that there's very little contrast. I mean, even for the slides, I'm talking about the slides, okay?
Okay.
The thing is, for a deafblind person, they cannot see what's going on in your presentation. It's even hard for a normal person like myself and my husband to see what's going on in your presentation.
Okay, because I have used images that we generally use in our talks, and to get them into a contrast and other things, only people from your group can help me. Otherwise, I will not be able to appreciate that.
Right, okay, I'd be more than happy to help in any way that we can.
Okay.
But right now, the slide that you are showing right now,
Mhm.
I can barely see it.
I have kept it deliberately like that. I did tell that because of my subject's privacy, I am not revealing many things. Okay. I have both a moral, ethical, and a legal implication. Particularly, this is an online talk. Okay. So therefore, I masked it deliberately, which I did tell in my talk. It's very difficult for us to show that, you understand, because it's somebody's life and I can't talk openly about it. This is a problem that we face in the field of medicine, okay? So when we give a medical talk, we always keep everything open. I understand your point right now, and that has been done deliberately. I did talk about this to Jerry, that I have this issue because I'm talking to a patient group and I have to be very careful. In fact, she wanted a copy to be circulated. I raised my concern because it is some information that I cannot reveal out because of my ethical restrictions. It is deliberately done to restrict the information. Today's world,
Right,
you can
and I'm
use
not
the
asking you to share any patient information. That's not what I'm asking at all. I'm just like, if you're doing a presentation, you can certainly take out the information of the patient and still be able to share relevant.
I'll give you an example. Okay, when I was working in New Zealand, we had a big family which we used for our study, and we took all consent, everything, and clearly told them this to be used in our study. And then when we published, these were not digital days. When we published in a hard print, their family, we don't know how, but they picked it up. They found it was in the journal. They came and discussed it with us. And since then, we have been jumbling the pedigrees. Okay. So that is why I masked the family tree completely so that nothing you can identify who it is. So I don't know if my own patient is sitting in this meeting. You understand? These are tricky things that we have to be careful in our profession when we do discuss these things. And I can't discuss very bluntly about the condition and the situation without showing the responsible slide. Okay. That is why these are tricky. I do understand your point, but we are legally bound and we have to be ethically careful about how we deal with these things. So therefore, we have issues on our side, and it's not that we are asking these things deliberately. But you should understand that. Okay. And there are the rules of genetics is much more stricter than the rules that we have in the field of medicine.
Sorry to interrupt. Yes, here. Yash said that he wants to log off now because he has some urgent work. So he don't need any ISL interpreter now. So he
Okay.
wanted us to be log off also.
This
Yes.
you're telling whom, please? This you're telling to whom?
So, he's telling to Gary that now
Oh.
I'm going to log off. So
This is Jerry. Saurabh, and ISL interpreters, please stay online so that you can continue signing, and others who want to view this recording can still avail of the information.
okay.
I would. Is that okay, Saurabh
Okay,
and
that's it.
Arwa?
Yes, he said okay.
Okay, and also for Diane and Jennifer, um, Aruna, I know it's difficult for Divya and to see the slides right now. Right now I am recording such that the slides are separate from the interpretation. So if the recording is done properly, you'll be able to view the recording with just the interpreters on it without the slides. So I hope that that will be some sort of a compromise. You may not be able to see the slides properly right now, and the interpreters might be small, but once I have the recording, you'll be able to see the interpreters without the slides.
Thank you, Jerry. Much appreciated.
All
Thank you
right.
so much.
Okay, so Saurabh and Arwa, please continue interpreting. And Dr. Kumar, please continue.
Okay, so Jerry, what I will do right now is that since the slides aren't taking that important role in the talk, since the audience has a vision and hearing issue, I would rather say that I will hold back my slide from this point and only give the talk. And we can have the interpreters much well blown up so that it makes much good sense. Will that be okay? Do you think?
Gary,
Uh,
Saurabh here. Please switch us. Arva is going to interpret now.
okay, Saurabh. Yes, Dr. Kumar.
Yeah.
Yes, if you can,
Arva, please turn on your.
if Dr. Kumar, if you can please
I'll stop.
unshare, stop sharing.
Okay.
So now we have the interpreters bigger. Now before you continue, let me first spotlight the correct interpreter.
Okay.
Just a minute. I'm spotlighting Arwa, and I am spotlighting Jennifer. Just a minute. All right. Is that better? Divya, can you see properly?
Yes.
All right. Thank you, Dr. Kumar. You can continue.
Okay. Thank you, Jerry. So I was talking about the last subject who has Stargardt's disease, and he had reading disability and almost lost his um, total vision. And I've seen him from very young age for a couple of decades, and he's been talking to me right from the premarital stage onwards, and now when he consulted me, came to the preconceptional stage. So before getting into the preconceptional stage, we actually tested him and his wife for the ocular issue of Stargardt's disease. So he had a classical Stargardt's disease as described in the textbook, where the cones are completely lost and so are the rods at the end stage. But his wife was perfectly normal as far as the vision is concerned. And when we did counsel him premaritally, he is from southern India and he chose his partner from northern India. So therefore, she was not related to his family in the first place. She was not of his own community in the second situation. And thirdly, she was not from the same geographical region. So he's from south and she's from north of India. So we tested him and then we found that from genetics he had a gene called as ABCA4. in which two of his exons is again a very large gene, exons 27, 47, there was a change. And these changes were pathogenic in nature, and that was positive of the condition. Okay. So this we call as compound heterozygotes. Now I'm using few medical jargons here. Okay. I have used them to make others understand and not keep it very simple, at the same time not too scientific, because you are people who could be a scientist, who could be an engineer, who could be a doctor. You can come up with ideas, okay, which we as scientists or as doctors may not think. Okay. So your bright idea you can discuss with a non-scientist or a doctor whom you're consulting, and that can, trigger and come out with something, novel. All right. That's the reason I kept my talk a little scientific in nature. It is not just to use some medical jargons. All right. So therefore, he had a change in the gene called as ABCA4 gene, which is one of the most common gene that causes Stargardt's disease across the world, and so it is in India. Now, we tested his wife, okay, and with my experience with the family tree, there was no connection between the male and the female in terms of blood, and also community and so on. And with my own practice, where we have not done genetic testing, almost 80% of my career, I was very clear in my mind that his wife will not carry any change in this ABCA4 gene like her husband. Okay. But when we tested, it so happened that she was carrying a likely pathogenic variant, which was a learning curve for me. Having been in this field of genetic counseling for the last 30 years, I was taken aback. Okay. Fortunately, we did this test, and when we counseled the affected person with Stargardt's disease, it was very clear that he wanted to test her. Okay. And therefore, when we tested, we found she had a change. So now we have a situation where the husband has two wrong copies, the wife has one wrong copy, and she has one right copy. Okay. Now the options are only two. The fetus can pick either of one wrong copy of the father and one wrong copy. If it chooses from the mother, it will be affected. Whereas if it chooses the right copy, it would not be affected. Okay, there's a 50-50% chance of the child being affected with the same condition. Okay. So these are situations where not only the patient and the family, but we as genetic counselors also get equally tensed. Okay. So a prenatal test was done. And when the prenatal test was done, fortunately, the fetus picked the right copy from the mother. Okay. So in 15 weeks, when we took the sample, by the 16th or 17th week of pregnancy, we told that this child will not be affected. This child is born, it's a female child. About a year and a half in the middle of pandemic, it was born. Second pandemic wave. And the reason I brought this to discuss today is that this is one of the solutions you can offer to your patients. Okay, and this was information. In fact, I gave this talk in the All India Ophthalmic Society meeting in Calcutta last month. And this was well appreciated by the physicians. They were totally unaware that we could do all this in India. Okay. leave alone whether they can be done or not. So therefore, these are awareness talks that not only the physicians should know about it, but also public and people who are affected should know so that they can talk and create an awareness amongst their own community. Now, with that, I come to the last part of my talk, which is the what are the types of therapies that people are looking at? Okay. Now, Usher syndrome, as I showed in one of the slide, has about 16 genes that are responsible for having Usher syndrome type 1, 2, and 3. Okay. Now, in the first intervention, which is called as DNA interventions, you can replace the gene, which is called a gene therapy, or you can correct the DNA letter change, which is called as gene editing or genome editing. And these two techniques are quite robustly done, and gene replacement therapy is very successful as far as RP65 is concerned. And Usher syndrome, Stargardt's disease, choroideremia, various other conditions are already in clinical trials, if I'm right, okay? Now that's about the gene therapy or DNA interventions, which is the first type. Now in the second type, there is something called as RNA interventions. Again, that is picking up much more speed and traction compared to DNA interventions because it's much more easier, less expensive. So RNA interventions, there are two types. One is called as antisense oligonucleotide therapy, and the second one is called translational read-through drugs and so on. So both these techniques are picking up, particularly the RNA interventions, where they try to do what is called as RNA editing. And that is much more effective and not too dangerous as a term called as the off-target issues, which is quite dangerous in genome editing. Okay. So therefore, one is DNA intervention, where we have two types: gene replacement therapy, gene editing. The second one is RNA interventions. The main one is called as the antisense oligonucleotide therapy or ASO therapy. And the next one is called as the cell therapy. Okay. And the most common one used in that is the stem cell therapy. Okay. So in that, there are different techniques involved where you can actually take a stem cell from the same individual and then correct that with some kind of editing and put it back in the individual. Okay, this is one of the safest one that you can do. Or you can actually take a cell from another individual and then you make it as a stem cell, which is very oriented towards retinal cell, and then you can implant it. But then that would need immunosuppression in the individual because the cell is not of self origin. It is a foreign cell taken from another individual and given into you, and therefore it's equivalent to doing transplant, kidney transplant or any other liver transplant and so on. So immunotherapy suppression, immunosuppression is mandatory in such kind of therapy state. But again, stem cell is not caught up, at least to the best of my knowledge, like the way DNA intervention, particularly gene therapy, is caught up with. But again, if you ask me the cost and factors like that, gene therapy is very expensive, unaffordable in fact. But maybe Nancy can throw better light about it for an Indian audience. And stem cell therapy may be much more cheaper, but then still we have not seen it to be very useful in ophthalmology. It's much useful in hematological disorders. Okay, at least that's the way it's perceived in the field of medicine. So that is about the therapy, and therefore I come to the end of my talk. I spoke about Usher syndrome at the beginning, and then explained what it is in terms of its effect in the retina and its effect in the cornea. And then I did talk about genetic counseling, which is my field that I work on day to day with my subjects. And in genetic counseling, we can help people in terms of diagnosis, or whether when they want to get married, when they want to have children, or they want to know what kind of gene the condition belongs to. Okay. But each time you do a genetic test doesn't mean it will result in a positive result. Okay. The chances of you getting the right gene and the right variant will be around 60%. Okay, 40% of times you'll be back to square one. And therefore, we don't recommend genetic test for everybody. So there are therapies, very promising. One is the DNA intervention, second RNA interventions, and thirdly cell therapies. Okay. So that's how the field is right now. But having seen the field pre-pandemic and post-pandemic, as somebody who's been in this field for more than 3 decades, I can see quite a lot of traction post-pandemic. The pandemic did actually create a big obstacle in the field of therapies, but then it has done good, I would say. Having gone to some other meetings connected with therapy, since last 2 years after the pandemic, I see that there's quite a lot of traction in therapies, and hopefully in less than a decade there'll be quite a lot of application and therapies that will be happening. Again, we have no idea what the cost will be, but certainly there will be sort of a crossroad we have faced during pandemic, and a new pathway will be found for therapies is what we all feel in this field.
Excuse me, doctor.
Yeah.
I'm sorry to interrupt you, but it's already at the end of our meeting time. So I would like to request you to wrap it up, if possible, in a minute or so.
I already finished.
All right.
Yeah.
All right. I would like to open the floor to 1 or 2 more questions, and after that, okay, there's a question. Can you please share the number of people affected by Usher syndrome in Tamil Nadu? That's one of the questions raised.
Well, I have no idea either. Nobody has that kind of data as far as I know. But if you ask me, if in my own clinical practice, how many Usher syndrome do I see in a month? Compared to retinitis pigmentosa, I've seen 3 retinitis pigmentosa patients in a day when I was working in Shankar Nethralaya. I'll see at least 1 or 2 Usher syndrome in a month. That's the difference in which I would see. Again, these are not patients from Tamil Nadu. I don't think such data is ever published actually. Even for retinitis pigmentosa, they would say that it's about 1 in 4,000 who are affected with RP globally. Okay, it's a conservative estimate. But Usher syndrome, I don't think such kind of data is available for our state, Tamil Nadu. Okay. It's not a big number.
Dr. Kumar, this is Divya's dad.
Okay.
Is in India anybody working on CRISPR technology for Usher syndrome?
Not that I know, sir. not that I know, because CRISPR is basically genome editing. I don't think anywhere in the world, Nancy might correct me if I'm wrong, anybody works with genome editing for Usher syndrome. Okay. Because the problem with genome editing is that it can't be, say, if you take stem cell, if you make stem cell, then it can work for anybody with, say, Usher syndrome. Okay, it's much more generic in nature. But genome editing has to be personalized. I'll have a mutation in, say, compartment 12. Another person will have it in compartment 15. Genome editing can't be the same for both of us. Okay, it has to be personalized. And therefore, I don't know if anybody is attempting, I've been searching for this gene therapy in Usher syndrome quite extensively. And Nancy connected me with this Sepal Bio group. Thankful to her. But I can find out and tell you, but at least at the clinical level, I don't think anybody is working. I don't know in the preclinical level if anyone is working on genome editing. But having said that, genome editing is the right way to correct a condition because you're correcting the DNA letter and the gene will start working. It's like putting back the carburetor. Okay. And the car will start working. So I don't know if anybody is working in that. Is my reply to you, but I can find out and then inform Jerry so that she can inform you.
Thank you so much.
All right, not at all.
This is Jerry. It seems like Nancy wants to say something.
Yeah, sure.
Yes. First of all, this is Nancy. This is Nancy speaking. Can everyone hear me? I'll look at the interpreters. Okay, they are signing, so I'm guessing that my audio is working. Dr. Kumar, thank you so much for this presentation. Every time we hear more about the research, I know for myself it sinks in a little bit more. I'll bring that car analogy around with me today. Very helpful. But there are a couple of things. I don't know of any CRISPR or any really large scale gene therapies that are taking place in the community right now. In case individuals don't know, we are having our conference in July, and our research presentations will be on July 20th. They will be available online. So if anyone wants to hear the latest updates on research, I'm sure Jerry can send out the link if you'd like to participate remotely and get those updates. Number two, the numbers were asked about. We at the coalition generally have taken estimates from researchers in various countries, in various studies, and we kind of generously say that between 1 in 17,000 births would result in a child having Usher syndrome. But again, as Dr. Kumar said, it's really just a guess. It's just a stab in the dark because we don't have good numbers. And what does help is the database that was referred to earlier, which is called the USH Trust, USH, and the word trust. It is about a 10-question survey, or if you will, and the Usher Syndrome Coalition maintains basic information about individuals who have Usher syndrome worldwide. We have about 2,600 people in it right now from 74 countries. And this data is used when researchers ask us if there are enough people to participate in a clinical trial, because we do assist with outreach and finding participants for clinical trials. So if you get into that contact database, you will be the first to know about research recruitment worldwide. I just wanted to give those 2 points for now. Thank you.
This is Jerry. Thank you, Nancy. Lata has her hand raised. And also, I'll have to say, Lata, that this would be the last question.
Can you hear me?
Yes, we can.
Okay, doctor, my name is Lata. I want to have some details about my son. He's around 50 and he has Usher's and he lost his, of course, he is deaf and he lost his vision almost completely around 4 years back.
Okay.
But since 6 months and since September, he's having a slight improvement.
Improvement
yeah.
No,
Yeah.
improvement of vision or hearing.
Vision.
Okay.
And to the point that he can now recognize a few rupee notes. He can work on the mobile phone. He can read the mobile phone, actually, messages. And so I just want to know whether it will ever improve this R3. Will it ever improve?
Well, basically, I have seen this and many people have reported also with our patients that we do see in our clinic. There is always a waxing and waning as far as vision is concerned. And again, scientifically talking about it, they say that there are some growth factors in the retina, and therefore certain parts of the retina gets revived, and therefore the photoreceptors become active. Since you said this, you kindly, I would suggest you probe more with your son. If there was any kind of lifestyle change that he adopted.
No, absolutely not. Absolutely not.
No, you better check it out. Okay.
Yes, because he's living with me.
Yeah. But
so
then
I know that.
It could be something with diet. It could be, with some kind of lifestyle practice, waking up or going to bed or, walking in the street and eating a particular food that he stopped.
No. Nothing of that sort. Absolutely no change. I know that.
No, there must be something. Otherwise, this won't happen, madam. So
Oh, I don't know.
you check with them. Okay, it can be some fear that he must have, gotten over, and therefore it could be certainly something very important because when you tell these things to scientists and patients, to doctors, then they can work from that. All right. So therefore, when I do have some of my patients who have reported such things, and I've probed into them, and you might find something. It might be some tablet that they would have started or they would have stopped. Okay, either one could actually result in this kind of change.
Okay.
So certainly, I have seen some people completely losing vision one morning. They give me a call saying they can't see anything, and then they regain it after a couple of days. Okay, these are very difficult to explain, but again, it could be something with growth factors in the retina and the retina getting revived.
Okay.
But that's a good news, actually.
Okay, thank you very much. Thank you very much.
Okay.
Just
Thank you, Jerry.
last question to Dr. Kumar, just quick.
Yes, sir.
Is L-histidine supplement helps for the Usher syndrome?
I'm not sure, sir. I'm not sure. There are a few studies which are related to amino acid replacement and few drugs also, which they do recommend. I've seen, and some clinical trials on the drugs are also going on with the NIH clinical trials. But I can find out both the questions you asked today. One is about genome editing in Usher, and whether it's preclinical or self study, I will let you know. and also about this L-histidine. Okay, I
Thank
will
you.
Jerry know so that we'll get the feedback.
Thank
All
you.
right.
Okay.
This is Jerry. Thank you so much, Dr. Kumar. We
You.
are 15 minutes over, but thank you so much for all the information. I know we struggled trying to understand the interpreters as well as the slides, but I think the question and answer at the end, that was worth it. One thing I need to mention also with Lata's question about her son had 17 years of treatment at Sridaryam, as well as something like a stem cell therapy, which they thought uh, did not work out. So, but that was a few years back. Whether that has anything to do with the sudden improvement, even little bit of improvement in his sight, um, it's not clear. But I just wanted to mention that because as a parent with a son with Usher, I want to hear good news that vision can be, improved. But you are correct. We need to research what
Yes.
has been done
Mhm.
to reverse this. What lifestyle changes has been done?
Okay,
Uh,
we will discuss about this if Madam Lata wishes, along with Jerry. We can follow up with that, and I can interact one-on-one online, and we can discuss, because even the stem cell therapy per se could have cost good or could have cost bad, and it would have waned away and the vision could have come back. It's difficult to guess these things, but therefore, uh, you know, it is something we have to discuss a lot, actually.
Are-
Excuse me.
Yes, Dr.
Can you,
Yes,
yes,
Verma.
and
Yes.
any information I can give you regarding Arun, I can give, there's no problem.
Yes, yes.
And
We
I'll be
will
happy
do
to share information and actually I'm
Certainly.
hoping that there'll be some improvement also, some more improvement.
Yeah, I'll come back to you through
Yeah.
Jerry, and then
Yeah, anytime, doctor. Thank you very much.
thank you, appreciate
all
it.
right. This is Jerry. I will set up that meeting because
yeah.
I'm also very interested.
True. Yeah.
But I have to say that we need to end the meeting now. I will, this talk is being recorded. I'm going to edit the captions and announce to the group when it is available for viewing. Thank you so much, everyone. I have to say good night now. Bye-bye.
Bye-bye, thank you.
Thank you.